Hi everyone. My colleagues made a fantastic job because now it's clear that the DNA is there in honey. And believe me that yes, indeed, it's there. So I repeat it like a third time here. And I'm really the guy who tried to convince you based on the data that yes, there is maybe one solution that maybe helps the beekeepers in the future. So, but let's go to the cases first. You recognize this young guy there? This guy? Bernard visited us in August, and based on his initiative, he brought us 30 anonymous samples. Among those 30 samples, there was one positive control. Positive, I mean authentic honey sample. We tested all the samples by total honey, like a honey total DNA sequencing. And what came out, actually you already know that 3 samples passed nicely as authentic honeys, and 2 samples barely passed with minor comments. So in total 5 samples were fine, and rest 25 samples failed. And we classify the samples as non-typical honey samples. Yeah, and those 29 were collected from supermarkets. So that is the real situation. The selection, let's say, the sample set was limited, but still it represented the real life. Next, then Bernard, Mario and other colleagues visited Finland, and they collected 56 labeled honey-like products from the supermarkets. So that reflects really the situation on the market. We analyzed those samples as well, and it wasn't an anonymous study for us, but still, and the outcome is here. 21 samples out of 56 passed nicely, but 35 samples failed. And we classify the samples as non-authentic honey samples. And if we calculate the percentage, it is more than 60%. The Finns were pretty sad about it. And the common feature there, the common trend, the samples what passed nicely, was that if you buy the honey jar from the supermarket, you can really check the label. And based on the label, it was clear that the producer was there, the address was there, the phone number. You can get touch to the producer. And all the past samples share the same information. And of course, Finns advertised that, yes, trust the label and trust the local beekeepers, and then basically there is a 0 chance to get fake honey. That was the clear message from this set. Another brave lady sits here, Lynn. She delivered us also 30 anonymous products from market, including 5 authentic samples from United Kingdom. Yep. And what came out? We nicely identified, Lynn's provided 5 authentic samples without any problems. And surprisingly, 1 sample out of 24 was authentic. The rest failed based on our analysis. And even the girls in the lab said that the honey samples tasted like lollipop. Because Estonians, we know how the honey should taste, but it didn't seem like a real honey at all. Because that is one of the standard procedures in our lab, the girls taste the honey. And it goes to the remarks. And as you see, 96% failed based on our analysis. You ask, is there any room to go? Can it go more worse? Should I continue? Really? Okay, I have one good example left. Wolfgang is there. Yeah, hi, Wolfgang. We made a collaboration with Wolfgang, and he delivered us 30 totally anonymous samples, again, including 5 authentic ones from Austria. Wolfgang can confirm it. And even I was surprised. We, of course, identified 5 authentic honey samples nicely, without any problems. All 25 samples failed. It means the failure percentage was 100%. Then I calculated is it the chance? You can calculate. Is it the chance to detect 5 samples incidentally among those 30? And the probability to hit the 5 samples incidentally, it's 0.0003%. In other words, yes, DNA helps to find authentic honeys and distinguish them from the fake ones, no doubt. As said, my name is Kaarel Krjutškov. I'm the head of Celvia laboratory. I'm an expert in DNA testing, molecular diagnostics, and DNA-based screening. I'm totally newcomer in food safety. That is not my specialty at all. But I guess I don't surprise you much, but there is no revolution in the food safety. That is just a time when the technology, what is known and applied nicely in the medicine, comes over to the food safety. There is no revolution. That is the normal technology transfer. And further I explain you how the DNA technology works in your field. I have published more than 70 papers. As I said, based on training, my diploma, there is a molecular engineering on my diploma. I work in molecular diagnostics and so on. My laboratory is a company, and it provides genetic screening for pregnant women to find the chromosomal diseases of fetus at very early stage of pregnancy. And what we did basically with a honey sample, we took the very similar technology, what we use in medicine, and we put it into the honey study. And that's why basically we use the most sensitive and most validated technology in the medicine, and we replace the source, we replace the human DNA with the honey DNA. If I see your testing systems in the food industry from behind, I see it's like 10, 20 or 30 years past. It means that 20, 30 years ago, when the screening started, then only the main goal was to find the Down syndrome during the pregnancy. And that was achieved by some kind of targeted method. So the pregnant woman was screened. She was Down syndrome negative. Everybody were happy, but still very many sick kids born. And there is no point to perform screening if you generate false negative results in the laboratory. In our medical world, it is absolutely non-acceptable if you provide false negative results. But as we saw from many samples here, not many, 4 samples here, the laboratories generate false negative results. And in Austria example, all samples were basically false negatives. In our world, it is totally non-acceptable. I would run bankrupt if I would release 1 or 2 false negative samples annually. I wouldn't survive that. But in some reason, NMR lab and other technique lab people, They are running their assays and techniques with a serious faces and working heavily, but still generating like false negatives. Come on, how it's possible? So, the genome is not only trisomy 21. It is not only Down syndrome. If the woman is pregnant, the family wants to be confirmed that there are no other chromosomal diseases as well, because everybody wishes to get healthy baby. That's why we do totally non-targeted DNA sequencing, and that touches the pregnant woman screening, because the nature provides very many chromosomal diseases what should be screened. And as you see, in addition to trisomy 21, there are like many other very nasty diseases screened. And that is possible with a highly sensitive DNA screening method. So let's come back to honey. We started the honey project 2020, and it was funded by the European Agricultural Fund for Rural Development. The aim was to develop the methodology for DNA testing. We knew that we can apply the technology from the medical sphere, basically. We adapted the technology a bit, and we are there. The project ended 2023, and we launched the honey DNA test April this year. And our project, in some reason, the name as Authentic Honey Meets DNA Technology, is a finalist for the ARIA Award 24 under the category Smart and Competitive Agriculture. So the election is still active. You can take the phone and give vote for us. But as an outcome of the project, of course, we developed the method. That was one aim, and we also composed the manuscript, which is published by our side, and it is under review. You can take a look, you can read it. It nicely describes the honey bulk DNA metagenomic analysis to identify honey biological composition and monitor honeybee pathogens. The adulteration question is not described there because initially it wasn't the research question at all. But nowadays we feel that yes, that is the main question. So, but the research tools, they provide very good tools to see the adulteration as well. So, if other peoples are studying proteins, polysaccharides, monosaccharides, whatever is in the honey, we study the DNA. And in this graph, the DNA proportion is the smallest. It's really the smallest. If we extract DNA, then we get nanograms of DNA, and that is enough. But as my colleagues already presented, the DNA contains all kinds of information, because the DNA is from this tiny honeybee microenvironment. So whatever honeybee touch or whatever happens in the hive, the DNA shows what is inside. It comes from the plants, of course, no doubt. There is the insect DNA, and not only the honeybee DNA itself, but of course other insects are represented. Fungi, that is, and yeast, that is one of the most significant part later on in the analysis. And don't forget the bacteria. You all know that human microbiota, the bacteria in intestine, they are important for our health. Actually, the same happens with honeybees. If you feed your honeybees with syrup, the same happens as basically if you eat only one type of food. you totally ruin your microbiota. And the same happens with the honeybees. And we see that through the DNA. And it's not a joke. Yes, we see beekeepers' DNA, really. And we see your cats, dogs, cows, whoever. And DNA viruses, don't forget that, not RNA viruses. So Michael made tiny mistake when he said that only some percent of the total DNA is, sequence DNA is informative. The percentage is 50, but it is informative enough to do like any kind of DNA studies. Per sample, we generate 10 to 20 million of DNA reads. That is the number of raw reads. And just after that, we ask computer basically to find the species where the reads belong to, where they have the perfect match, basically. And there is no limitation because we use all published genomes here, published and collected and generated by research community. The next stage, yes, we can characterize the plants, and the same. There is no limitation because if the researchers and public institutions provide the full genomes of different plants, they all are there up and at high quality. So that is public database. And now I basically answered to the criticism that is it like some kind of Baltics or like a too European specific? No, it's not. We maybe just think that we as a Europeans sequence more species, but it's not correct. The Asian species are much more sequenced than in Europe. It's much better covered than European species. And based on DNA, basically we can see all DNA-based pathogens and parasites from the honey DNA. You are familiar already with such kind of nice plot. Yep. But what you didn't see from Mikael's presentation, you didn't see the sector of bacteria. And you didn't see the yeast and fungi. You didn't see the DNA viruses. Because if you target some species group, like a plants, then you see the plants only. And if you skip the fungi and yeast, you do not see the fungi and yeast. And if you skip the bacteria, you don't see the microbiota of honeybee. If you take the honey bulk DNA sequencing, I mean the total DNA sequencing without any manipulations, you really see the full content of species what are in. And the beauty of this kind of graph is that you can zoom in and go closer and closer, like a deeper and deeper, what is there in the honey DNA. You can now guess, is it possible to manipulate with such kind of DNA profile? Like if you add some kind of DNA powder? No, there is no chance. Because we do not take account only the number of species here, but we see the balances. Because in case of authentic honey, the balances are nicely there. You cannot add one type of DNA and hope that you cheat the researchers. No, there is no chance. Because it is more than clear that if you add some kind of pollen here or bacteria DNA, the balances changes drastically. So this kind of sector is like a DNA fingerprint, let's say so. But that is also like a digital fingerprint. We didn't agree totally with Michael, but as you see, we are thinking in the same way, and we are basically using the same symbols. The same type of graph, the same QR code, and so on. Because if you do such kind of DNA analysis, then it turns the honey production basically and tracking the product more transparent. You can go back using such kind of QR code and see such kind of report. Okay? The laboratory procedure is relatively simple and robust. First, there is a total DNA extraction. It is automized. Then we prepare the DNA library for DNA sequencing. Here we generate this 10 or 20 million of DNA raw reads. And after that, we start the data analysis. First of all, we ask the question about the honey DNA profile. There we use all genomes database what is available. We get the profile there. Then we visualize it using for the botanical composition and honeybee pathogens and parasites composition. What are the balances there? And after that, we make the comparison to ask about the adulteration and origin. The origin is not so straightforward because we share the plant species here in Europe, and if the Finns, for example, bring the honey samples for testing, then the machine says that that is Estonian honey. So plants do not ask the border of the country. But in case of the adulteration, yes, it's really powerful method. Actually, the data analysis, we try to understand what happens there, because if you have like thousands of species and you have to keep in mind the balances between the different organism groups, there is no chance to do the analysis without computer. So that's why we have trained the computer models that is called machine learning or AI, which is more sexy word nowadays. But the computer really helps us to analyze the data because the data is so complex and so multidimensional. That's why. But I still try to explain how it looks like. So for example, I created a lot of green dots here as an authentic honey database, let's say so. So for the machine tries to find the midpoint of the database. The midpoint is somewhere here, and you also see the borders somewhere there. So that is the authentic honey database. In good scenario, if you have authentic honey, yes, it bumps somewhere here. The one model, for example, says the score is 96, so it's really inside the group. It's the perfect scenario, no doubt. Works perfectly. And do you want to know the scores of Austrian authentic honey samples? Yes, they are on the board. The scores were 100, 99, 99, 99, and 65. And as Wolfgang confirmed, the 65 was acacia honey. We do not have this piece so much in Estonia and in Baltics, but you have it, but it's still nicely inside the group. And it's like a perfect shot here, like basically tens only. And that's why you can play with the plants and replace the plants inside the honey. But you do not cheat or do not change the outcome of the analysis. So the computer really sees the patterns, how they behave, and changing the plant composition is the normal variation there. So that is the meaning. Of course, there is chance to get the borderline outcome. For example, here, if the score is 55, for example, then the specialists take a look as deeply as possible and makes the final decision because we know the markers of adulteration. And we make the final decision, and then the specialist decides that, yes, that is the decision. Is it in or out? But now comes the most famous guys here. You can check the original video of those 4 samples. I don't name this as honey samples, just 4 samples. please go to the Clean Up the Honey Market page in the LinkedIn, and you find a video, 15-minute video about those 4 stars. The name of the video is The Truth Behind Honey in Global Market. You see, polyfloral honey, UK grade, nice. Polyfloral honey, NMR grade, nice. And then some kind of rubber honey and floral honey. Actually, that is the same composition based on DNA analysis, just differently labeled. And what the computer said? Computer said that the scores of such kind of product, 0, 1, 1, 3. And that is like a dimensional difference from the authentic honey group. If somebody mix this product with authentic honey, it's still here in this dimension. If you add like a smaller proportion, yes, probably it moves closer to the authentic one, but there is no point to put 1 or 20, like a 10 or 20% of such kind of product, because you don't win much. You win much if you sell this kind of product as a honey, but it is not authentic honey. So that is the outcome of the analysis. It is really black and white. So take-to-home message. As I said, I'm expert in DNA technology in medicine. That feeds my laboratory and people. And the honey project is my side project, really. It doesn't basically bring in anything. It just takes money. But it is fun. And I really see the difference between the disciplines. In medicine, we wouldn't survive if we would release the false negative outcome constantly and making like a serious face for testing the samples and reporting false negatives. That is not acceptable. But in your world, it happens, and you all make like a serious face that, yes, that is the life. No, it is not normal life. I don't know why that happened, but somehow, for me, that is absolutely non-acceptable situation. If we take 25 samples from supermarket and they all fail, that is 100%. There is, it cannot go worse. In Estonia, by the way, it is not published, but in Estonia, the fake honey percentage is 30%. We come out with a news soon, but there is still authentic honey in food store. And still really good situation in Finland as well with this 40%. But in Central Europe, the situation is really catastrophe. Next message is there is no revolution because it was just time who brings the honey total DNA technology to your world. It is just a question of time. This time Celvia presented it, and that is the same scheme that if you wish to study the full picture, then you have to study all chromosomes. You have to study the whole DNA, what is inside the honey sample. And that reveals the proportions, distinguish the fake honey from the authentic one. And the final message is that I really taste that that topic is interesting. That's why it is clear that Celvia applies testing laboratory ISO in 2025. We collaborate with local authorities in Estonia. The ministry and other guys support us. So yes, we are going that way. The total DNA sequencing will be the standard soon. It doesn't happen in 3 months, but it happened in 2 or 3 years. And yeah, that is no normal progress. Just adapt or die. That is my message. That's all from my side.